In order to provide maximal yields, plants need to be provided with a suite of nutrients. Nutrient deficiencies, stresses such as cold or extreme heat, drought, or pathogens cause massive losses in crop yield every year. The origin of many of these problems often lies underground.
The root is the physical anchor of the plant, but it is also the organ that is responsible for water uptake and for the uptake of mineral nutrients such as nitrogen, phosphorus, sulfate, and many trace elements. If we want to develop sustainable approaches to producing high crop yield, we need to better understand how the root develops, how it takes up this wide spectrum of nutrients, and how it interacts with symbiotic and pathogenic organisms. To do this, we need to be able to explore roots at the microscopic level.
For obvious reasons, studying the biology of roots has always been more challenging than the study of aerial parts of the plant. Since roots are usually hidden underground, they are not easily accessible for microscopic studies. Removal from soil causes severe damage to the root system and is thus not a good way to study their behavior. One solution to make roots more accessible is to grow them on gelled, or as shown in this example, in hydroponic media.
Growing plants in gelled media or in hydroponic media has been very successfully used in many root studies. But with these methods, it's still very difficult to study roots in microscopic detail and over long periods of time. To get as close as possible to the growing root and avoid any physical stress from preparation for imaging, we build a platform that allows us to grow and image roots and at the same time allows us to control and modify the microenvironment of the roots with very high precision and speed. This platform we called